Today we're going to talk about transport across the membrane, so how things get into and out of the cell through the plasma membrane. This is broken up into two lessons, so this is just part one, and it's the passive transport. So essentially we're looking at diffusion and osmosis. The next lesson is going to talk about active transport. So with passive transport, the movement of a substance across membrane is happening without any energy from the cell. So no ATP is being used. So this is driven by diffusion. So diffusion, remember, is the movement of a substance from a high concentration to a low concentration. And this is done because when you have a high concentration of molecules, whatever it is, whether it's sugar or water, when there's a lot of it, they're bouncing off of each other. And they're going to cause each other to move away from each other because they're hitting off each other. And they're going to move to an area where there's less of them because they will be bouncing off each other less. So that's essentially how diffusion happens. So let's look at our diagram here where we have our semi-permeable membrane. So some things can pass through this membrane. In this case, we have molecules of dye that can pass through. And there's a high concentration on the left, and there is a low or no concentration of dye on the right. And as they bounce off of each other, they will hit some parts of the membrane that can go through, but there are particles that will pass through the pores and get into where there is a lower concentration. This is going to create a net diffusion, a net movement of molecules into the area where there was a lower concentration. This will continue to happen until we have what is called equilibrium, where you have the same concentration of the particles, in this case the dye particles, on both sides of the membrane. So there are some factors that affect the rate of diffusion. So we looked at factors that affect enzyme activity, and it's actually kind of similar in the sense to things that affect rate of diffusion, specifically temperature that we looked at in the lab that was so successful. This is recorded in 2022. So future students might not be getting that lab. So the temperature, as temperature increases, the movement of molecules, because they're going to have a higher kinetic energy, so they're going to bounce off each other more. So a higher temperature is going to cause a higher rate of diffusion. The concentration gradient, so the difference in the concentration on one side versus concentration on the other side. A higher concentration gradient is going to cause a higher rate of diffusion. When the concentration is the same on both sides, like I said in the last slide, movement between the two still occurs. So it's not like everything just stops moving. It's just the net movement stops. So there's still going to be movement from one side to the other, but you're not going to see any overall change in concentration. So this is called dynamic equilibrium, or we just say equilibrium. Okay, membranes, as I said previously, are selectively permeable. So that means that they let some molecules diffuse through.
At this time, I kindly ask teachers.
Some molecules can diffuse very easily through the membrane, whereas some are unable to pass without some form of transport to assist. So looking here, this is a great diagram to highlight to help you remember. So small nonpolar molecules can just pass through. So something like oxygen, carbon dioxide, or nitrogen can just pass right through. And we can imagine the severe importance of carbon dioxide and oxygen to just be able to come in and out, which allows for respiration. Small uncharged polar molecules, most of them can just go through, but some will be kept out. For example, we have water, and we have glycerol and ammonia. Large uncharged polar molecules, most of them cannot go through. So things like glucose, for example, have glucose-specific transporters to get them into the cell. And then ions, charged ions cannot get through and need specific transporters to get them into and out of the cell. And we're going to look at some examples of these in our next lesson. So factors that affect how easily a molecule or ion can pass are based on size. So for example, the glucose and sucrose, and charge, and that's where we were looking at ions. Okay, so looking at simple diffusion. So this is the ability of substances to move across a membrane unassisted, so they can just pass right through. And again, we're looking at these hydrophobic molecules like oxygen, carbon dioxide, and nitrogen. And then most of these small uncharged polar molecules like water and glycerol can just go through. And again, large or charged, tend to not be able to get through, and again, would need an active transporter to help them. So looking at facilitated diffusion, so it's not just going right through the plasma membrane, but they would need some sort of channel to allow them to go through. So demands for many polar and charged molecules can be too high for passive transport to be allowing enough into the cell. So they need something to help them just get through quicker. And so we have these channels, if you will, to help them pass through. So this is called facilitated diffusion. So the facilitated transport of ions in polar molecules happen through this membrane protein. Often it could be one protein, or it could be more than one protein, so those would be called complexes. So protein complexes means that there is more than one protein bound together to create this channel. It's still driven by diffusion. So the important thing to remember between this lesson and the next lesson is we're still working with diffusion. We still have molecules moving from in this case, a high concentration to a low concentration. Tomorrow, or in our next lesson, we're going to see how it's actually going from a low concentration to a high concentration. So it's actually going against this concentration gradient. So today, just talking about diffusion, high concentration to a low concentration. So we have these transport proteins. So this is the nice way that textbooks like to draw our transport proteins, but we've seen they definitely are a lot more complicated than that. So this is an integral membrane protein, so a protein that passes through the lipid bilayer, and it provides a pathway for the molecules to cross. And we have this hydrophobic section, and we have the hydrophilic channel that's produced. So it's still aqueous, and it's still allowing the hydrophilic molecules to go through. Okay, so just looking at different channels, so that was considered a channel protein. So it's a hydrophilic pathway in a membrane that enables water and ions to pass through. We have specific ones that we look at in this course. So it's a very common one is a sodium-potassium channel, and we're going to look at that in a little bit more detail in a little bit. We also have a calcium and a chloride channel. So most ion channels are voltage gated. So they switch between open and closed by changes in the voltage across the membrane, or by binding molecules. So we have a ligand gated, so that's how you say ligand. So ligand gated channel. So if we have sodium attached, then it closes the channel, and the sodium can no longer go through. We also have mechanically gated ones, which are really neat. They have some sort of substrate that would be attached to it, and will actually just mechanically close off that channel when it's not needed. There are ones that are always open, and then as I mentioned, we have our voltage gated ones. And we're going to talk a little bit about membrane potential in the next slide. Okay, so it's really important, especially for Unit 4 for us to understand that there's a difference in charge on the inside and the outside of the cell. So membranes tend to have an electrical potential across them. So it's almost like a battery. So it's negative on the inside of the cell and positive on the outside of the cell. And this really, really helps the cell do certain functions, and we'll talk about that later. But it actually is really helpful for the cell to be able to do this and have this. So it allows for the cell to act as a battery, and this provides power for the operations in the cell. What we're going to see in Unit 4 is how neurons get to use this difference in charge across the cell to transmit signals throughout our body. So again, just a close-up on the voltage-gated ion channels, because so many are of this type in our cells. So they're going to open or close depending on the voltage across the membrane. So again, if we have positively charged or if there isn't much of a change, then they might open or they might be closed. So it really depends on the type of channel. So all you need to know is that it's based on the change in the charge on one side versus the other side of the cell membrane. There also might be a case where it binds a solute and it'll either be open or closed. Often used in nerve conduction and in muscle contractions, which we don't look at the muscle contractions in this course, but there is definitely an interest in that field in university. So the opening and closing of the channels are triggered by changing ion concentrations. So again, depending on the difference in the charge, so positive versus negative, across the membrane, the ion channel might open or close. Okay, we also have carrier proteins, and these are extremely helpful in getting substances through the cell membrane. And again, this is going to create a passageway, and remember that they are still, the solutes are still moving along their concentration gradient. So they're going to go from a high concentration to a low concentration. We just need a little bit of help sometimes, maybe because of the size of the molecule. So how these work is they will bind to their specific solute. So they're going to be solute specific. So for example, we could be looking at one like glucose. That would bind specifically glucose, and or tryptophan. So tryptophan, remember, is an amino acid, and it is a precursor for which neurotransmitter? I will ask you tomorrow. So the transporter will bind its specific solute, and it's going to actually change shape. So that's really interesting about a lot of these transporters. So in this case, we have the solute gets bound to its site where it would bind to the site where it would bind, and it changes shape, which allows it to essentially get dumped into the cell. Okay, carrier proteins changes shape when the solute binds, so that's really important. So that is the distinguishing feature between a channel, which just acts as like essentially a tunnel for the solute to go through, versus a carrier protein, which is going to change shape and then release the solute into the cell. Okay, as mentioned earlier, the most carrier proteins are very selective about which solute they will carry. So in the last example, I was talking about glucose or tryptophan. It wouldn't, a carrier protein would not transport both of those. It would transport just glucose or just tryptophan. So for example, transporters that transport glucose are actually unable to transport fructose. And we know how similar glucose and fructose are. They're isomers of each other. This allows for tight control of what gets into the cell or other organelles. And this is really important. So the cell can have very strict regulation of what is coming into the cell and staying in the cell. the types of transporters in plasma membranes, and on the other outer membrane of the mitochondria depends on the type of cell and growth conditions. So again, depending on the needs of the cell, it's going to have different transporters in the membrane of the plasma membrane or in other organelles. So with osmosis, it's the movement of water across the membrane. And it's very important in the medical field, and we're going to look at some examples tomorrow in a case study in class. So there's 3 types of solutions, and they're always in compared to another solution. So we have hypotonic, and this is where we have a lower solute concentration. We have a hypertonic solution where we have, in comparison to another solution, it's going to have a higher solute concentration. And then we have isotonic where you're going to have 2 solutions and they have the same amount of solute in both of them. Okay. Water is going to diffuse or move to an area of low solute or a high water concentration to a high solute or a low water concentration. So it's always important to remember you're comparing either the solute or the water concentration. So typically we talk about a solute concentration. So we would say water is going to go from where there's low solute to high solute concentration. And these brackets, these square brackets, means concentration in the science world. Okay, so water would move from an area of low solute. So if there's a little bit of sugar, for example, in a solution, the water would move to where there's more sugar because it's going from an area of high water to low water concentration. So there's 2 ways to look at it. It just, as long as you communicate yourself properly, you'll be correct. So let's look at these examples here before we look at them in class. So here we have a sucrose solution, and we put it into 3 different other solutions. Okay, so we have one solution compared to whatever's in these beakers. So we put it in distilled water. The water is going to go from a no solute whatsoever in the distilled water into where there's a high solute concentration. And so we're going to see the swelling of this sac, it's a bag, swelling of the sac because water moves into where it has a high solute concentration. And we can see this is what would actually happen to our blood cells if we end up with like a really, really high salt solution in our blood. And we'll see why this is important in our case study tomorrow. Similarly, if we put this in a 10 mole per liter solution, so much more sugar on the outside than inside the sac, you're going to see water leave the sac and go into the beaker solution. And this is where we would end up with shriveled up cells. Now, if we put this 2 moles per liter sugar solution into a beaker that also has 2 moles per liter, there's still going to be a movement of water, but it's going to be a net movement, and we don't see any change in the shape of our cells.